Hey guys, today we're going to talk about 1.2 cell ultrastructures. So when we say ultrastructures, we're really talking about the organelles in the different types of cells. So first things first, there are different kinds of cells. This is kind of a throwback to freshman year, but let's just review. So cells are divided into categories based on their structures and complexities. And the major categories that we're going to talk about are prokaryotes and eukaryotes. In the picture, you can see prokaryote on the left. It looks like a bacterial cell. And on the right, you see a typical animal eukaryotic cell. Now, just looking at these pictures, you can tell there's quite a big difference between the two, and we're going to spend some time talking about the parts and pieces of each. Let's start with prokaryotes. This is understanding number one. Prokaryotes have a simple cell structure without compartmentalization. These simple cellular structures include the nucleoid region, containing naked DNA, 70S ribosomes, the cell wall, plasma membrane, cytoplasm, pili, and the flagellum. Okay, so prokaryotes, right? They came first. These are the first types of cells to evolve. Their genetic material occurs in plasmids, little tiny rings, and it's not contained within a nucleus. They have a pretty simple structure without compartmentalized organelles, and they were the first evolved life forms. So when I say plasmids, I mean like they have little rings of DNA, little circles. So they don't have, they have dual-stranded DNA, some have RNA, but instead of being wound up into chromosomes, right, or long loose strands of chromatin, they literally just have like these little tiny circles of DNA. So our prokaryotic structures, well, first they have a cell membrane. Anything that is a cell has a cell membrane, right? And that's what surrounds the cell and controls what travels in and out. It separates the cell from the rest of the environment. Inside the cell membrane, we have cytoplasm, which is the liquid contents within the cell membrane. It's composed mostly out of water, but there's some other stuff in there too. And this is the site of metabolic reactions within the cell, things like photosynthesis or respiration or both. Now you have a cell wall, which is located outside of the plasma membrane, and it's what provides the structural support to the cell itself and gives it its shape. Now, not on here, but not in the text, but here in the drawing, you'll see capsule. Capsule is another layer on the outside of the cell wall, and it's what the pili and other kind of extracellular attachments are attached to. Internal structures include ribosomes. Now, when we say 70S, it's a specific type of ribosome. You can think of it as, how like cars have different models, right? And they get like more complex as time goes on. Or think about engines. You have like a regular engine and then you have like a turbo engine. The 70S ribosome is a ribosome that is an earlier version of what eukaryotes have, which is the 80S ribosome. Now, remember, ribosomes used to be their own, well, we've talked about this a few times, but we'll talk more about it in 1.5. Ribosomes used to be their own kind of independent thing, which is kind of interesting. But yeah, so the ribosome, 70S, you can think of that as a version of ribosome that's specific to prokaryotes. So if you see an organism and somewhere it's noted that it has a 70S ribosome, it's for sure a prokaryote because all eukaryotes have 80S ribosomes. Next, you have the nucleoid region. Now, it would be, if it was enclosed in a membrane, it would be the nucleus, but there's no membrane there. So we just call it the nucleoid region. And this is where all the DNA kind of hangs out. Then we have some external structures that you should know, like cilia, that help assist in mobility as well as moving extracellular materials around. And then there's the flagella, which is a long whip-like tail, and it's used as a motor for movement, right, for swimming. Now, we also heard about pili, which is not shown here, but those are those hair-like projections that help bacteria adhere to other surfaces. So I'm gonna go back, and these guys, these hairs, this isn't cilia. It kind of looks like cilia, but it's not. These are pili. This is to help the bacteria hold on to, you can think of them as like little fingers. They hold on and they grasp whatever the surface is that they want to hold on to. It's important for bacteria that are infectious, like pathogens. They want to like hold on to a surface like the inside of your mucous membranes in your nose. That's what those pili are all about. Okay, let's go back. Okay, so this is application number 1. Prokaryotes divide by binary fission. Now, it's basically just like mitosis, but like really simple. We don't give it lots of complicated names because they're prokaryotes and we don't want to. So we just call it binary fission. And this is asexual reproduction, where the cell duplicates its genetic material before splitting its DNA and cytoplasm in half, and then the daughter cells are identical. So if you look at the diagram, we have our cell with our one chromosome, which because it's a circular loop and it's a prokaryote, we call it a plasmid. Sorry if you hear all this stuff in the background. I'm visiting at my aunt's house and there's like, family junk going on. Okay, so that one chromosome is a plasmid. That plasmid is going to duplicate, right? So now you have 2. And then you're going to get division of the plasma membrane and the cell wall, and the division of the cytoplasm, which is called cytokinesis. And eventually you have 2 identical daughter cells, right? So this is just like mitosis. No kind of exchange of genetic material, no variation. Evolution this way tends to be extremely slow because it only comes from a random mutation within the DNA. You don't get any kind of like mix up of genes from different individuals. So evolution tends to happen pretty slow. That's why we were stuck with prokaryotes for, a few billion years before eukaryotes evolved, because, slow process, less chance for change ups. Okay, so now let's talk about eukaryotes. Remember, you are a eukaryote. So prokaryotes are simple organisms, and they were the original life form. So when we're talking about prokaryotes, we're talking about here, right, bacteria. Eukaryotes eventually evolved later on. So here we have our original common ancestor, right? We have descent, we have a common ancestor, we split. These guys are prokaryotes. And then we have these guys, which become archaea and eukaryotes. And when we think about eukaryotes, remember, eukaryotes include plants, fungi, and animals. Okay, eukaryotes are organisms that are complex and that have compartmentalized structures called organelles. Most notably, their DNA is contained within a nucleus. They are so loud. Eukaryotic organelles include the nucleus, right? Now, the nucleus is an envelope, the nuclear envelope, and its purpose and its job is to protect the DNA on the inside. Now, this membrane is a double membrane, meaning it's a phospholipid bilayer, just like the cell membrane itself, right? So it's got the phospholipid ping pong ball with 2 tails, and then you have that other layer, phospholipid bilayer in the tails, right? So it's just like a regular cell membrane. That nuclear membrane is made of a phospholipid bilayer. Now, there are holes in that, and that's to let mRNA pass in and out. DNA never leaves the nucleus. I'm going to say that again. DNA never leaves the nucleus, right? The whole point of the nuclear envelope is to protect the DNA. So DNA replication and transcription, all that stuff happens inside the nucleus. Okay, next is the cell membrane. And like we said, that's the phospholipid bilayer. So there's one layer and there's the second layer. It's a bi-layer. It goes both ways, literally. See, top down, bottom up. Haha. Okay, so the cell membrane is extremely important for a couple of reasons. One, it's what defines the cell, right? If there's no division between the environment and the internal contents of the cell, then the cell's not a unit of life. So you need that barrier there. Now you notice it's not just phospholipids. There's a bunch of other stuff in there. And we're going to spend a whole subtopic 1.3 that deals with all the different things that build this membrane. You can think of it as like the skin of a cell. And just like your skin isn't, just skin, there's like pores in there and there's glands in there and there's all sorts of stuff that make your skin a functional organ. There's all sorts of molecules that make the membrane a functional organelle in the cell. It's responsible for letting things in and out. You can see some facilitated protein channels right here. We have some signaling molecules here. Your cells don't have like eyes and ears and stuff like that, right? But they still have to know what's going on. They have to exchange information with the environment around it. So these little molecules that stick out, they're signaling molecules. They pick up information from the outside and relay it inside. The cell membrane is probably, I think, if I had to pick, I'd have to say it's like probably the coolest out of all the organelles, or like the most functional. It's the most important one, because without it, nothing gets in, nothing gets out, no information gets in, no information gets out. And if something happens to the cell membrane, the cell dies. So yeah, I'm voting cell membrane for president. That's my story, and I'm sticking to it. Okay. We have the 80S ribosome, because remember, we're talking about prokaryotes. Ribosomes are important. These guys are protein synthesis sites, the sites of protein synthesis. So they're like, I don't know if you guys watch Fraggle Rock or whatever, but like these guys are the doozers. And if you don't know what I'm talking about, I want you to Google Fraggle Rock doozers, because that's what the ribosomes are. They're the doozers. They're awesome. Okay. Some ribosomes hang out in the endoplasmic reticulum, the rough endoplasmic reticulum. That's what makes it rough, because you see all these little dots on it. Now, the endoplasmic reticulum itself also has a job. Its job is to package proteins and send them out to vesicles, to the Golgi apparatus, right? So you can think of them as like the ribosomes are the workers, the endoplasmic reticulum is the factory where the proteins get made. The ribosomes go to work there and they hang out there, and the endoplasmic reticulum will package them and send the proteins to the Golgi. And then the Golgi is like the FedEx. So when I say vesicles, what I'm talking about are just little pieces of cell membrane, right? And their job is to surround and transport materials. Now, they can be transporting materials to or from the endoplasmic reticulum, or to and from the actual cell membrane. So it's important to remember that vesicles, right, they're made out of the same lipid bilayer. They're made out of the same stuff as the cell membrane and the endoplasmic reticulum. So they're all made out of like the same magic carpet that becomes the surface of the cell, or it becomes like a pillowcase that wraps stuff inside of it and transports it back and forth between the endoplasmic reticulum, the Golgi apparatus, and the cell membrane. It's all made out of the same thing. So you know how like when you blow bubbles and sometimes the bubbles will stick to each other? The cell membrane is a material just like that. They can stick together, they can pull apart and make bubbles, and their membranes can fuse right back together again. They're really cool. Together, these three things are called the endomembrane system. So I should probably put that somewhere, right? I'll write it up here. endomembrane system. And by the way, this is understanding number 2. So let's start with, we'll start in and go out. So we'll start inside the nucleus. So your DNA is in the nucleus, right? And your DNA holds the information to make proteins. Well, mRNA is going to take that information and take it to the ribosomes in the Golgi, not the Golgi, the endoplasmic reticulum, right? So DNA, and then the mRNA is going to take that information because DNA can't leave, right? So the mRNA is going to take that information to the little ribosomes. Now, the little ribosomes, which are these things, right? They're going to do their thing and they're going to produce through transcription and translation. I guess this is translation, right? Through translation, they're going to create their proteins that they're supposed to make. And those are those little tiny blue dots. And so as those proteins get made, they get pushed out. And then you see here at the end, this is some of that endomembrane. And it's going to pinch off and form a little bubble. And it's going to carry that stuff to the Golgi. And so this is a vesicle, right? Now that membrane is going to merge and those proteins are going to get dumped in here. And then the Golgi is going to be like, oh, I recognize this. I know where it goes. So it's going to repackage it into more vesicles, which is made out of the same material. Again, so this material here, like this membrane is the same as this membrane is the same as this membrane, right? And then it's going to package it and it's going to send it. And then through exocytosis, that same membrane is going to fuse with the cell membrane and spit out the protein, right? And then the circulatory system will carry it wherever it needs to go. And then the same thing happens in reverse. So say, a cell needs to take in a particular protein for a certain thing or a particular substance, right? It's going to do the exact same thing in reverse. It's going to come in, form those vesicles, right? Deliver it to the Golgi. Golgi is like FedEx. It's going to figure out whatever is being brought in and it's going to send it where it needs to go. Okay, so we have the rough ER, which is like the factory. And then we have the Golgi, which is like FedEx. And then we have export out of the cell. Coming the other way, we have endocytosis, which brings stuff into the cell, so import. You have, it gets delivered to the local FedEx office, and then the FedEx office tells it where it needs to go. That is the endomembrane system. Okay, now let's talk about why multicellular life is possible. So these two guys right here, these guys are the whole reason that life exists in multicellular format and on land, right? So first, I know it says mitochondrion first, but we're going to start with chloroplasts because chloroplasts is like just the coolest thing. Chloroplasts are responsible for photosynthesis, of course. These are the tiny little things that are responsible for converting light into chemical energy. So without this, all that energy from the sun wouldn't be harvested. Now, these guys actually used to be their own prokaryotes. They used to be their own thing, their own living organism. And through endosymbiosis, right, they became engulfed in another prokaryote cell. And then the descendants of that fortuitous merger became plants. Now, additionally, that same thing happened with mitochondria. So mitochondria also used to be their own prokaryotes. They were their own thing, and they were engulfed by another prokaryote. And instead of being digested and eaten, they formed a mutual symbiosis, endosymbiosis. And just like these guys are responsible for collecting energy, collecting and storing, mitochondria are the opposite. These guys are responsible for releasing that energy. And when we say releasing, We gotta jump back into the realm of some chemistry for a minute, and let's go back and think about respiration and photosynthesis. Now, I know you might be thinking, like, I haven't thought about that stuff since like freshman year. So let's just think about this. So energy from the sun is chemical energy, right? And it's stored in the form of glucose, right? And glucose is C6H12O6. Well, that's great, except for that your cells can't actually use glucose. Your cells use ATP instead. You can think of it as like money, right? So glucose is a form of energy or a form of currency that your body uses to store energy, but it can't spend it. It's like money in the bank. You can't spend it unless you have it in your hand, right? So you have to think of a way to get that money out of the bank. And that's what mitochondria do. They are responsible for converting that energy that's in glucose and turning it into a form that they can spend, which is ATP. Next, we have some organelles. Now, these are only important during mitosis and meiosis. Probably spelled that wrong. Microtubules, they are the threads that play an important role in lining chromosomes up during cell division. And then centrioles, these guys are responsible for anchoring the microtubules on the sides of the cell. They'll become more important when we talk about mitosis, but just as a review, these things are important organelles, but they're only temporary. So you don't see them all the time. You only see them during mitosis or meiosis. Okay, so now let's talk about some of these specialized organelles. Vacuoles are similar to vesicles in that they are made out of that same membrane, that phospholipid bilayer, same thing as the Golgi, same thing as the ER. It's that magic carpet that can fold and bend and become a pillowcase to carry stuff, or it can become the outside of the cell, or anything in between, really. But you can think of a vacuole as like a giant storage space. Now, plants have very, very large vacuoles, and that's for a very good reason. They can't move very much. Like they don't move from place to place. So they need to be able to store materials. So, for instance, like water. Animals can just get up and walk somewhere else and go find water. Plants can't. So their cells need to be able to have large storage areas to maintain supplies. Now, those supplies could be water, right? It's going to store water in itself for when there's not a lot of water in the external environment. It could also be to store glucose, to store starches. It just depends on where the cell is. So if this is a cell that's in fruit, a lot of that vacuole storage space is going to be used to store starch, sugar, because that's what it is. It's a cell in the middle of endosperm or the tissue of a fruit. Or if it's in the stem or if it's in a leaf, this large vacuole is probably going to store water for use in photosynthesis. And just so that we're clear, this is the actual vacuole. Now, animals do have vacuoles, but they're much, much smaller because they don't typically need to store very much. Um, Animal cells are pretty much on demand. They get what they need on demand. They don't store very much. They use stuff, they get rid of it. Plants also have that very rigid cell wall. This is something that sets them apart from animal cells. And that's because plants don't have bones, right? So like each of their individual cells has like its own little cytoskeleton. And if you remember from our macromolecules, it's made out of cellulose, right? It's a structural carbohydrate. It's those sheets of carbohydrate rings, beta sheets, right? So they're structural. They make, wow, that's really bad drawing. You can basically think of it as like a brick wall, only the brick should have 5 sides, not 4, but whatever, you get what I mean. Okay, So the cell wall is rigid. It's a structure that's found outside of the cell membrane in plant cells, and it provides structural support. Now there is a line between um, two plant cells between their cell walls, and it's called the middle lamella. It's just like the border between two cell walls, two different cells. Um, you don't really need to know the primary cell wall, secondary cell wall. You do need to understand that the cell wall is separate than the cell membrane, and when you're doing drawings, your cell wall needs to have two lines, and then your cell membrane also has to have two lines to indicate the thickness of each. Okay, so you've learned this like a million times since middle school. Plant cells contain chloroplasts, plant cells have large vacuoles, and plant cells have large walls, right? That's what makes them different. Now let's talk a little bit about microscopes and how we use them to visualize cells. This is understanding number 3. Electron microscopes have a much higher resolution than light microscopes. So electron microscopes, they use electrons and shoot them as a beam or as a wave over the surface of a substance, and they literally destroy that substance. As those electrons bounce off that surface, an image is created using the basically the point where the electron beamed back, kind of like a radar gun. So like when cops use a radar gun on you, they shoot like a radar beam at you and it bounces back to their gun and that tells them how fast you're going. Well, electron microscopes work much the same way. So you have a surface and then that electron is beamed down and then it bounces back. And as the beam moves, it bounces back at different angles and an image is created in response. Now we need electron microscopes to look at prokaryotic cells because prokaryotic cells are so very small. Look, we're looking at, this is 100 nanometers, right? Here we have 10 micrometers. 10 micrometers. Look, this is 4 micrometers. So like this is probably like what? 1, 2, 3, 12 micrometers across. Super small. And that makes sense because they're pretty simple. They don't need to be big. They don't need to fit all those organelles, right? Okay. At this point, you should be able to identify the following organelles. Well, you should be able to identify all of them, but in particular, number 2, 10, 3, 11, 5, 13, 6, 14, and 9. Let's see if you got those right. Well, you'll see in your questions, I guess, yeah? Okay. Now you should be also able to identify the organelles in the plant cell below. And how about I give them some numbers? So we'll give this a number 1. We'll give this a number 2. We'll do this a number 3, and then inside number 4. Let's do these little black things. Number 5. Let's do, we'll do this thing, number 6. We'll do this whole thing, number 7. Yeah, I think we'll leave it at that. Hopefully you can identify those. Okay. Part C, micrographs and drawings. You know what? Let me pause the video here and I'll start another drawing or another video.